Hello and welcome. On behalf of CME Outfitters, I would like to welcome all of you and thank you for joining us for the third in a series of 3 CMEO snacks titled Investigating Efficacy and Safety of Novel and Emerging Dengue Vaccination Options. This CMEO snack series is supported by an independent medical education grant from Takeda Pharmaceuticals in US. My name is Marco Aurelio Safadi. I'm an associate professor and currently I'm the head of the Department of Pediatrics at Santa Casa de São Paulo School of Medical Sciences in Brazil. And I have the pleasure to be joined here today by my distinguished colleague, Dr. Inés Esquilin Rivera. Dr. Esquilin Rivera, could you please introduce yourself?
Hi, Dr. Safadi. Yes, my name is Inés Esquilin. I'm a professor at the Department of Pediatrics of the University of Puerto Rico School of Medicine and the director of the Pediatric Infectious Diseases Division at the university in San Juan.
Okay, excellent. Well, the objectives of today's program is really to look at the data that we currently have on terms of safety and efficacy of the vaccination options going into the clinical practice. Well, in our last program, we discussed it a little bit and we talked about what dengue is and the current situation in US. Could you please remind us, Dr. Esquilin Rivera, of the current situation of dengue in US?
Yes, as you know, dengue virus has 4 different serotypes, the serotype 1, 2, 3, and 4, and it's the most important mosquito-borne illness, and is transmitted by the Aedes aegypti and the Aedes albopicus mosquitoes in the endemic areas. There is an FDA-approved vaccine available, and there are other 2 promising emerging options that we can talk about. It is projected that the risk of dengue in the US is going to increase, and it will include more areas in the southeast region of the country. So it is important to be aware that probably in the future we will be seeing more cases of dengue in the US.
So that's currently the situation all over the world. We are seeing an increasing number of places, regions at risk of dengue. This is really an issue we are facing. So now that we know a bit about the presence of dengue in United States, what currently we have available in US in terms of prevention of the disease with vaccines, and what do you currently foresee in terms of prevention in the near future, Inés?
We do have one approved live attenuated chimeric dengue vaccine. It was approved in 2019 and recommended by the ACIP in 2021. It includes the 4 dengue serotypes, and it was approved for children between the ages of 9 and 16 only if they live in endemic areas and have laboratory evidence of prior dengue infection. Then there are 2 candidates, the Butantan TB03 vaccine, which is in phase 3 clinical trials and is similar to the NIH-developed TB003 vaccine. Is also a live attenuated recombinant vaccine and seems to be very promising. And then the Takeda 003 vaccine, which granted priority review last year and the endpoints of clinical trials were met and is under review for FDA approval right now.
Very interesting. Actually, we have just licensed the Takeda vaccine here in Brazil a couple of months ago. The vaccine was licensed. So we do have some new perspectives in terms of prevention. Thank you so much for that introduction, Inês. So let us talk now initially of the first of the dengue vaccines, the CID TDV vaccine. Could you expand a little bit more on how is this vaccine composed?
Yes, this is the vaccine that is currently approved. It has the yellow fever 17B backbone, and then the genes, the E and PRM genes isolated from each serotype are introduced in this backbone. It is a vaccine that, as I said, is indicated only for people living in endemic areas, not for travelers. And this vaccine can be given to children only if they have evidence of prior infection with dengue. So if a person is going to be vaccinated, testing has to be done prior to the administration of the vaccine.
Excellent. And let me know, please, what is the current perception and what do we currently know of the efficacy data of the vaccine? What have you learned about the efficacy of this vaccine against the several endpoints associated with dengue disease?
Yeah, the vaccine has an overall efficacy of 82%, and it was approved because of the data that revealed that it was not only efficacious in preventing hospitalization and severe disease, but also disease at all. So the vaccine proved to be safe and very efficacious.
Okay, so in summary, the vaccine proved to be efficacious and around 60% against virologically confirmed dengue. This was the endpoint that reached these results, correct, Dr. Inês?
The vaccine efficacy against severe disease and hospitalization and disease at all meet the FDA standards of efficacy. It depends on the clinical trial data. The one that got the FDA approval was the trial that demonstrated an efficacy close to 80%.
Yeah, you usually see higher estimates of efficacy for the most severe endpoints associated with the disease. And we also observed a very efficacy estimate against each of the 4 serotypes. We also observed lower efficacy against serotype 1 and serotype 2 comparing to what was observed against serotype 3 and 4. And also very clearly, we observed lower estimates of efficacy for the younger age group, particularly young children in the studies. And maybe for me, the most important result was the really lower efficacy observed in the seronegative subjects that enter initially in the study. We know that the vaccine is not currently licensed for naive persons, but we clearly observed a lower estimate of efficacy among these individuals. And what about safety, Inês? What can you tell us about the safety data that we currently are aware of this vaccine?
The safety data was very good. The most common adverse events associated with the vaccine were local reactions, headaches, and myalgias. It was very safe when used in individuals that had evidence of prior dengue infection. And I think that probably that's the most important point. But in those that were naive to dengue at baseline, the vaccine resulted in an increase of the risk of severe disease. So that's what the clinical studies demonstrated, and that's the reason why the vaccine should not be used in patients that are naive to dengue, that have no experience with the disease in the past.
Excellent. Well, thank you so much, Dr. Inés, for reminding us of the efficacy and safety data. But let me now move to what I think really matters when a vaccine is licensed, which is the real world data. So I really like to see how those vaccines behave in the real world setting, not in a clinical trial, but also when we currently use. And I'm happy to share some results from Brazil, where a study has been conducted in a region that implemented the vaccine. So let me just share with you the results of this study that was conducted in the state of Paraná, in the southern of Brazil. This was a case control study. It was done in the 5 most populous cities of this state, and the vaccine was used, and then they were able to really evaluate the effectiveness data. And regarding these results, the only variable that was significantly associated with effectiveness in the matched stratified analysis was effectiveness against serotype 4. Against serotype 1 and serotype 2, the case control study was not able to demonstrate any effectiveness. There was no serotype 3 circulating during the study. So the only serotype that the vaccine proved to be effective among the 3 that were circulating was serotype 4. It's important to highlight, Inés, that this is a region in Brazil with a low seroprevalence, and this study was done before that recommendation of only giving the vaccine to a person that was previously infected. So it's very likely that we had several persons that were naive to dengue and were vaccinated at this time. It's also important to acknowledge that the main serotypes that circulated during the study were serotype 1 and serotype 2, which we do know that are 2 serotypes that the vaccine proved to be less effective, less efficacious comparing to serotype 3 and serotype 4. So these are, I think, are important topics to be acknowledged and considered in the interpretation of the results, as well as the low coverage. The vaccine never reached good coverage in this region, but these are the results we do have. So let me now, I would like to discuss another emerging vaccine option, and you mentioned it as a promising vaccine, and I do agree with you. It appears the preliminary results appear really promising. It's a unique vaccine, as Butantan dengue vaccine is being developed in my country, in Brazil. It was licensed to the Butantan Institute by the National Institute of Health in US. So let's talk a bit about this vaccine. Here we can see how the vaccine is constructed. Actually, we have 2 candidates, TV003 and TV005. The difference is regarding the amount of the dengue 2 component in the vaccine. Butantan has manufactured a lyophilized version of this tetravalent live attenuated vaccine, and the efficacy evaluation is still ongoing. All the components of the vaccine, all monovalent components, have a dengue virus genetic background. So this is clearly a difference regarding the CID-TDV, which has the yellow fever virus, the vaccine virus as the background. Here we have the dengue virus genetic background, and they share a core attenuating nucleotide deletion in the region of the viral genome, yielding replication deficient attenuated virus. So we see in the slide the composition of the vaccine with the 4 monovalent components. Regarding efficacy and safety, Inés, a single dose of this TV003 formulation vaccine proved to confer sterilizing immunity in 100% of the subjects that were vaccinated in a challenging study 6 to 12 months later. A single dose of either TV003 or TV005, both formulations, they both induced seroconversion to the 4 dengue serotypes in 74% in the case of TV003 and in 90% in the case of TV005 of flavivirus naive individuals. So these were seronegative adults that received both formulation. We also observed very interestingly that a large proportion of the subjects that were vaccinated developed a rush. So this rush was observed in approximately 70% of the subjects that were vaccinated. And last but not least, regarding this Butantan dengue vaccine, we don't have the final results of the study. The study is still ongoing, but the Butantan Institute provided very recently a press release when they were able to share preliminary results of the study that is being conducted in Brazil. And the results that they shared provided an estimate of the efficacy. So it's important to acknowledge that in Brazil, only serotype 1 and serotype 2 circulated during the study. So the data I'm sharing is the data associated with serotype 1 and serotype 2. So far, we did not have the opportunity to evaluate in Brazil how the vaccine behaves against serotype 3 and 4. There are some studies being conducted outside Brazil, in Asia, where we will probably be able to really have results against the other serotypes. But the efficacy data we currently have available show it that against serotype 1 and serotype 2, efficacy was 89% against dengue 1 and 69% against dengue 2. And these were efficacy against virologically confirmed dengue. So very promising results we anticipate in ACE. Let me now move to you, and I would like to listen from you. What do you have regarding the Takeda vaccine, the TEC003? Please share with us an introduction of the vaccine, Inés.
Sure, the results that you just presented are very promising, as are the ones with the TAK003, the Takeda vaccine. which granted a priority review in November 2022 and is going under FDA approval right now. Recently, it was filed for FDA approval. This vaccine has been on clinical trials for the last 12 to 13 years. Since 2010, and it has been evaluated for the prevention of dengue disease caused by the 4 serotypes in patients 4 to 60 years of age. It was approved, has been approved in many countries, including Indonesia, Europe, Argentina, Brazil, and Portugal. And this vaccine is a 2-dose vaccine that does not requires serological testing.
This is very important. I think this is also very promising vaccine to be used in places where dengue is a burden. Can you please expand a little bit on the vaccine? How is it constructed, the composition of the vaccine? Please tell us a bit more about this, Ines.
This is a live attenuated recombinant vaccine that was constructed using the dengue 2 virus backbone as the one you just talked about. And then it expresses the E and PRM proteins genes of dengue 1, 3, and 4. So that's the way it was constructed. It induces cellular immunity. The genes, the PRM and the E genes, are the ones responsible for the neutralizing antibody response. And it's a very novel vaccine construction and different, completely different in a way from the one that is approved, that has FDA approved, the first one we discussed.
Excellent. And Ines, let us know about the results of the pivotal phase 3 trial that has been conducted, and what do you have about the trial design? Tell us a bit about this, please.
This trial specifically was designed for children aged 4 to 16. A total of more than 20,000 participants were included, and they received this new vaccine or placebo in a 2-to-1 ratio assignment. A total of 91% of the participants completed the 4.5 years after the second dose, and that was the safety set. There was a primary endpoint, which was the overall efficacy, and some secondary efficacy endpoints, which were specifically efficacy by serotype and compared with the baseline serostatus and severity. So this data was recopilated. It was very important that there was an active febrile illness surveillance during the first 3 years of the study, and a long-term efficacy and safety data that is still going on. So it was a well-designed study, and I had the opportunity to participate as one of the investigators in the study, and it was a very well-designed study.
Oh, that's amazing. You were one of the investigators of this study, and obviously it brings you a more familiar perspective of how the vaccine works. And Ines, share with us initially those primary objective results, and also which are also important, the secondary objects of the initially short-term efficacy.
Yes, 12 months after the second dose of the vaccine, the efficacy for virally documented disease by any serotype was 80%, which was very good. 18 months after the second dose, the efficacy of virological confirmed disease leading to hospitalization was 90%. So this was a very good efficacy result. The primary and most of the secondary efficacy endpoints were met when there was enough dengue cases in the area for analysis. So there was also efficacy data that was secondary endpoints, and it was related to the dengue serotype and how good was the vaccine preventing disease according to serotypes and according to the fact that the patients were naive or not naive at the time of the vaccine administration. And 18 months after the second dose, the vaccine was 76%, had 76% efficacy in those patients that were seropositive at baseline, and was 66% efficacious in patients that were seronegative at baseline. So there was a little difference between the ones that were positive to prior exposure and not, but overall, the vaccine was very efficacious in prevention, irrespective of the serostatus. When you look at the data on the specific dengue serotypes, for dengue 1 was 60, almost 70%. For dengue 2, it was 95% efficacious. For dengue 3, it was a little bit lower, and for dengue 4 too. For dengue 3, 48%, and for dengue 4, 51%. So that data comes from that clinical study, and in terms of the different serotype data, it is important to mention that the efficacy against dengue 3 in baseline seronegative participants was not demonstrated, and that a robust assessment on dengue 4 serotype in the seronegative was not possible due to the low incidence of this disease by this specific serotype. What we do know is that the vaccine was very efficacious against all 4 serotypes in those patients who were seropositive, and for dengue 1 and dengue 2 in baseline seronegatives.
Excellent results, Inés, and obviously we also want to know about the long-term data. So these were data related to 12 months and 18 months after the second dose, but we know that Takeda has recently shared those results in the 4.5-year data after the second dose. Could you please summarize what were these results and what information could we get from these long-term efficacy data?
The long-term efficacy data was very good too. The rate or virologically confirmed disease or hospitalization due to dengue was overall 61% efficacy documented. And then according to the different serotypes in seronegative, positive and seronegative individuals, the vaccine was able to prevent disease in a long-term follow-up up to 54 months after the second dose.
Excellent. We clearly see that we have a trend of lower efficacy among seronegative subjects, naive subjects, when they received the vaccine. These appear to be very consistent with all the data. And we also have observed specifically for those seronegative subjects, for those naive subjects, that protection against serotype 3 was not observed. The opposite was demonstrated among the seropositive, where efficacy was really demonstrated against the 4 serotypes in the long term. And as you mentioned, against serotype 4, we still have very limited data to really have reliable interpretation of how the vaccine may work against serotype 4 specifically. Was very limited, the circulation of serotype 4 in those places where those trials were conducted. Is this also your impression, Inés?
Yeah, definitely. We'll see in the long term what will happen, because probably they are considering the administration of an extra dose of the vaccine, of a third dose, in order to deal with the low immunogenicity specifically to dengue 3 serotype.
Oh, very interesting. This booster dose is something that they will look to see if it's really needed and when it's needed. It will be very interesting to see these results in the long term. But it's very important that so far we haven't seen any safety issues among those naive subjects that were vaccinated. I think this is a very important message, and brings me to the next question, is exactly regarding safety. What do you have to share with us in terms of safety, Inés?
The safety data is what is very promising, because the major concern with the approved vaccine was specifically safety in those persons that were naive to dengue. The cumulative rates of serious adverse results events were similar in the vaccine and the placebo group recipients. It was very similar and very consistent with the expected medical disorders in the study population, and no deaths were considered related to the new TDV vaccine. So in general, it is important to say that the safety profile of this vaccine is very good.
Excellent. Thank you so much, Dr. Esquilin Rivera, for teaching us so much today. I hope our audience has learned a lot about current and emerging vaccine options for individuals. Let's review our SMART goals that we hope our audience has accomplished from participating in this program. The first is how to really identify FDA-approved and emerging vaccine options for prevention of dengue and limitations associated with these potential options for individuals at risk of dengue. The second is to apply clinical trial data to clinical decision-making when determining eligibility and vaccine choice for individuals seeking prevention methods for dengue. And last, scrutinize clinical trial data pertaining to efficacy and safety of vaccine options, both currently approved and soon emerging, that meet current unmet needs of individuals. This CMEO SNAC is one of a 3-part series. We hope that you will take advantage of all of the short and focused activities in this series. So again, let me thank you, Dr. Esquilin Rivera, for joining me today, and thank you to our audience for participating and providing the best care for your patients. Thank you.
Thank you.